The upper portion of the GI system will be explored here today, moving from the mouth to the pyloric sphincter. The primary anatomical features include the mouth and associated structures, such as the tongue, salivary glands, and teeth, the esophagus, and the stomach. Together, these organ and structure perform the functions of food ingestion, propulsion or movement of food, secretion of mucus, water, and various enzymes. Ultimately, mechanical and chemical digestion occur in the upper GI tract. When we take a closer look at the tissues found in the GI tract, we find 4 specialized layers. From the inside outward, they are the mucosa, the submucosa, the muscularis, and the serosa or adventitia. These are concentric layers of tissue of varying thickness, and each layer contains sublayers. These layers change in different portions of the GI tract, suited to the primary functions occurring in those areas. In the upper GI tract, we have mucosal functions of facilitation of transport, protection of deeper tissues, and glandular secretions. In the upper GI submucosa, blood vessels and lymphatics perfuse and drain the GI tract, while neurons and connective tissues provide control, feedback, and adhere the layer to deeper layers for stability. The muscularis layer is exactly what it sounds like: muscle and the associated nervous tissues to control function of those muscles. These may be voluntary striated muscle or involuntary smooth muscle, and forms a circular layer and a longitudinal layer. Lastly, we have the serosa or adventitia, while contains primarily connective tissue, holding the tract in place within the body and providing protection from damage. The mouth is the typical entryway into the digestive system. Primarily, the oral cavity serves to masticate or chew food and mix it with saliva. Therefore, the teeth and tongue are important for this purpose. 32 permanent adult teeth and the muscular tongue also help with speech. As food is masticated, particles move around the oral cavity and stimulate taste buds, bundles of neurons that detect salty, sour, bitter, sweet, and savory or umami flavors. In addition, food particles can stimulate the olfactory nerve as odor and contribute to satisfaction of eating and preparing the GI tract for salivation and gastric juice secretion. 3 paired salivary glands are found in proximity to the oral cavity. The submandibular, sublingual, and parotids glands produce about 1 liter of saliva a day. Saliva is mostly water, but also is composed of mucus, sodium, bicarbonate, chloride, potassium, and salivary alpha amylase. This helps initiate carbohydrate digestion in the mouth and stomach. With a pH of 7.4, saliva helps neutralize bacterial acids and serves as an immune system adjunct via mucin, or IgA antibody, which reduces rates of infection. The salivary glands are innervated by the SNS and PSNS. Beta stimulation decreases salivation, while acetylcholine stimulates secretion of saliva in preparation of the digest part of rest and digest. Swallowing is a sophisticated process where food is seamlessly transferred from the oropharynx to the stomach, assisted by both coordinated muscle actions and gravity. This process begins in the oral cavity, where the tongue propels the food into the pharynx, leading it to the esophagus, a hollow tube through which the food travels down to the stomach. Swallowing involves a series of muscular movements that seal the trachea and bronchus by lowering the epiglottis. The esophagus, measuring roughly 25 centimeters, 9.8 inches in length, is safeguarded by the upper esophageal sphincter, UES, which shuts to block the influx of air during breathing. As the process of swallowing progresses, the food passes through the US into the esophagus, where rhythmic muscular contractions, known as peristalsis, guide the food bolus downward. The top third of the esophagus and the oropharynx require conscious effort to function and are common sites for swallowing difficulties or dysphagia. The remainder of the esophagus operates involuntarily, governed by the vagus nerve and the hormone gastrin. Eventually, the food bolus reaches the lower esophageal sphincter, LES, also known as the cardiac sphincter, which relaxes to admit the food into the stomach. The LES serves a protective role by keeping stomach acids and enzymes from backflowing into the esophagus, which can cause damage. When the LES malfunctions, it can result in a variety of medical conditions. This is a hollow organ composed mainly of muscle, found just below the diaphragm. It stores food during eating, secretes digestive juices, mixes food with digestive juices, and propels partially digested food, called chyme, into the small intestine. Upper portion of the stomach is the fundus, followed by the middle, or body, and then the antrum, or lowest portion. The stomach is protected by the LES before the fundus and the pyloric sphincter at the end of the antrum. The stomach features a substantial mucosal lining that guards against infection and shields the tissue from its own digestive enzymes and acids. When the stomach is empty, this mucosal layer is pleated into structures known as rugae. The stomach's absorptive capacity is minimal, primarily limited to certain fat-soluble compounds, including alcohol and aspirin. Supply to the stomach comes from both the sympathetic and parasympathetic divisions of the autonomic nervous system. Digested food mixed with digestive juices from the stomach into the small intestine. When in a resting state, the stomach holds approximately 50 milliliters of fluid, maintains minimal tension within its walls, and undergoes only mild contractions. Swallowing causes the fundus to relax and receive the food bolus from the esophagus. This is stimulated by the nerves gastrin and cholecystokinin. Once food enters the fundus, peristaltic waves move toward the antrum. This is activated by the hormones gastrin and motilin, along with parasympathetic nervous system, PSNS, activity, and is suppressed by sympathetic nervous system, SNS influences, and the hormone secretin. The peristalsis mixes food and digestive juices, forming chyme. Takes several hours and consists of forward peristalsis and retropulsation of chyme. Each time chyme hits the pyloric sphincter, a small portion leaves the stomach. Eventually, antral contractions open the pyloric sphincter and allow chyme to flow out. The rate of gastric emptying can be influenced by volume, osmotic pressure, and chemical composition of the chyme. Larger volumes empty faster, while solids, fats, and non-isotoic solutions slow the rate of emptying. Some disease states alter gastric emptying rates as well. The intricate balance of hormones and enzymes in gastric juices is finely tuned. The production of these secretions is triggered by various stimuli, including the mere thought, smell, or taste of food, the physical expansion of the stomach, and the presence of substances like histamine, ghrelin, somatostidine, and acetylcholine. Additionally, the ingestion of proteins, caffeine, and alcohol can also prompt gastric secretion. Parietal cells located within the gastric pits of the stomach lining release hydrochloric acid. This acid plays several critical roles. It helps dissolve dietary fibers, acts as a bactericide to eliminate harmful bacteria, and transforms pepsinogen into the active enzyme pepsin. Chief cells within the gastric pits secrete pepsinogen and pepsin, which are proteolytic enzymes responsible for breaking down proteins into smaller polypeptides. The activation of these enzymes is prompted by an acidic environment, while an alkaline environment leads to their inactivation. Mucus is secreted by mucus cells situated in the upper regions of the gastric pits. These cells generate a protective coating that forms a barrier between the stomach lining and the gastric juices. This barrier is fortified by prostaglandins and bicarbonate, which help neutralize the effects of pepsin and the acidic gastric environment. In the stomach, a range of enzymes are produced that aid in the digestion of carbohydrates and fats. Concurrently, hormones play a regulatory role, either stimulating or inhibiting digestive enzymes, modulating appetite, and controlling acid secretion. Intrinsic factor, a crucial substance produced by the chief cells, facilitates the transport of vitamin B12 from the small intestine into the bloodstream. Similarly, gastroferrin, produced by parietal cells, binds to iron and assists in its absorption within the small intestine.